Nobody else could either, which is quite disappointing. Although in culture it does have some growth effects. And these are some early papers done by Dr. Kahn with the FGF receptor in liver cells in culture. But one of the findings that he found in 99, in one of his studies there, was with the receptors, he found that the FGFR4 isotype is the resident FGFR isotype in hepatocytes. There's no other of the other three types. They're usually partitioned in the non-parenchymal cells or in the stroma, we would call it. So receptor 4 we became very interested in because not much had been done about it. So a postdoctoral, a student actually, one of my students that now is a professor at Xiamen University in People's Republic, picked up this problem with the mouse models. He asked the question, if you have an FGFR4 knockout mouse, we knocked out the gene in the mouse, which didn't have a developmental phenotype, does that affect liver regeneration? Well, to his disappointment and ours, it had no effect on liver regeneration or liver damage. So this again suggested, well, maybe FGF receptor R4, the main receptor there in hepatocytes, it doesn't play a role in liver cell growth, even though we believe it should be a growth factor. So this is very disappointing to a student. This was what he did his first project, no phenotype. So what are you going to do? He's worked for a year or two, and is he going to change projects or drop the project? But Chindong Yu was a very clever graduate student. And what he did was to, when you do a partial hepatectomy in a rodent, you lift up the liver lobes, and right under those lobes is the gallbladder. Well, he noticed that even though it didn't get effect when he was taking out part of the liver, the gallbladder was odd. It was little in these knockout mice. So he was a very perceptive guy, and in our laboratory I let him run with this and say, okay, why is the gallbladder small in these knockout mice? And this is just an example of what he saw. These are the gallbladders he saw there. And he did a long series of experiments cell biologically, looking for cellular homeostasis ideas. There's really no effect on the gallbladder. Gallbladder's normal, liver's normal, environment's normal. But since gallbladder's involved with bile acids, he analyzed cholesterol and bile acids in the blood, and lo and behold, to our surprise, for the first time, the knockout mice had an elevated level of fecal bile acids in the bile acid pool. So this, for the first time, led us to start asking a series of questions of what is the mechanism, and I'm not going to tell you all about that, but it opened up that actually, with a long series of experiments, that FGFR4 represses CYP7A, the major rate-limiting enzyme of conversion of cholesterol to bile acid. And so for the first time, and this was in 2000, his paper here, this suggested that FGF signaling is important in a major metabolic circuit, cholesterol to bile acid metabolism, in a system where we could never show an effect of cellular homeostasis, like a growth factor, like it historically was in development. And this opened up now a whole idea that we are in today that has just exploded, is the discovery of specific FGFs and the receptors we've been studying as an endocrine system, just like classical hormones. First in metabolic homeostasis, and then soon after it was found that FGF23, a mutation in it, gave a phenotype in mice that was a abnormality in calcium and phosphate metabolism. And this opened up the idea that FGFs can regulate phosphate calcium metabolism. The factors involved with our early R4 experiment was FGF 15 and 19. Other groups picked it up, and they found that they got the same bile acid phenotype, but they also got effects on obesity, adiposity, and related to lipid and glucose metabolism, as well as with another subset of FGFs, FGF21. Now, FGF 15 and 19, these are misnomers. They're 15 is the mouse counterpart of the human 19. They were given a different name early on, a number. These three FGFs are what we call now endocrine FGFs. And if you look at them, they're characterized first on the heparin binding interface. Here's a classical cellular FGF, FGF2, basic FGF. They have a very low basicity and a lot of acidity on the heparin binding interface. So they all have low affinity for heparin in contrast to most of the other FGFs, or heparin sulfate. And also, it turned out that they have reduced mitogenic effects, but some overlapping activity in pathways to the cellular FGFs. So this opened up a long series of questions of how does all this work. But before I go into that, the several other groups, Brown and Goldstein and Dallas, who are the grandfathers of cholesterol, and the Dallas group confirmed our results with R4, and they proposed in one of their papers with a series of experiments that there is a FGF15-19-R4 axis, an endocrine axis, as shown in this paper here, that is a major controller of bile acid, cholesterol, bile acid metabolism in the organism that we totally unsuspected, that we opened up in 2000 with the R4 knockout. We sent them the R4 knockout, and they extended this and substantiated this and proposed it even in more great detail. Okay, I need to go back one. There we go. So from all this activity, and I'm giving a lot, I'm not going to give you all the reasons and the background on this. This has caused a global idea somewhat proposed by David Moore. The endocrinology picked this up very rapidly, by the way, and because it's very exciting for endocrinology, to discover new hormones, new endocrine factors that we hoped all the way back with in the early days that FGF might be, that we were looking for new hormones, but it was a tissue growth factor family. So this is the paradigm now that has emerged, that different low molecular weight steroid or low molecular weight molecules that act on steroid receptors that are transcription factor receptors are mediated through FGFs. And they are passed on down to the common 4 receptors, which we'll talk about in a moment, to give you these diverse endocrine effects. Here we have fatty acids act on PPAR alpha to give you energy homeostasis. Bile acids, like I talked about, go down through 15-19 and R4. And vitamin D acts through the vitamin D receptor, which controls 23 in the relevant tissues, and goes on down to control phosphate and calcium homeostasis. Well, I was happy to see all this because we were participating in this early pathway here, but we proposed this back in 1980 for prostate, for classical steroids, by the way. And so I added this to this figure. These, most of the classical endocrinologists have not put this in their scheme yet, even though we talked about it way back, that possibly steroids would go through steroid receptors, and that FGFs would be affected like the paracrine FGFs, FGF7, 9, and 10, which we worked with in our laboratory as well. And they would be a mediator in this same paradigm here for cellular compartmental homeostasis. So, and that we do still believe is partially true. So, we then asked the question, since we got a endocrine phenotype of cholesterol to bile acid synthesis, I'm not using this mic, by the way. I think you can hear me fine. So, can everybody hear me without this? Oh, you're recording. Okay. Okay. You may have missed some of it. Okay. I need both hands. Okay. We then asked the question: Does FGFR, since FGFR4 is a key player in cholesterol bile acid metabolism, does it play a role in lipid glucose metabolism and what we call metabolic syndrome? Now, most of you, if you're clinicians, you've heard of metabolic syndrome. Metabolic syndrome is being looked at much more now because it's correlated closely with obesity, which is a rising epidemic worldwide, particularly in Western countries, and is catching hold, I think, in Asia as well. These are the parameters of the metabolic syndrome. If in the clinic, if you have 3 of these, you've got metabolic syndrome, and you should be watched closely and probably take preventive treatment or so, because all metabolic syndrome is associated with a wide variety of cardiovascular diseases, side effects, and hormonal endocrine abnormalities, as most of you, if you're clinicians, do know much more than I do. Now, one parameter that is not a clinical parameter of metabolic syndrome is you get fatty liver that correlates with obesity. If chronic obesity gets fatty liver and causes chronic fatty liver disease and eventually fibrosis, and even gives rise to some hepatomas, which is a major symptom in chronically obese people. So here's a summary here of what I just said about the reason metabolic syndrome is of great importance and interest to us nowadays. Is I don't know if any of you seen the Super Size Me movie. This guy went on McDonald's food for 30 days, and he gained weight. He got fatty liver. His blood pressure went up, and it tells what happened to him here. And this illustrates here a cartoon here of what's going on in our schools that is a problem. And this guy can't buy his Cokes with sugar in there because he can't put his money in the machine. And this guy probably does have fatty liver, you know. And here's the consequences of non-alcoholic fatty liver disease, which is associated with metabolic syndrome. And as I said, this is not only happening in where I am in Western countries, but also in China and Japan, I believe as well, is the statistics of obesity and chronic obesity and associated diseases is rising rapidly. So another graduate student of ours who's now in a postdoc at UCLA picked this problem up, and he asked the question of, does FGFR4 in these mice, does it affect parameters of metabolic syndrome? And indeed, what he found after screening all these different parameters, that the mice were, as we'll show a little bit here, the knockout mice had more white adipose tissue, not brown fat, but white adipose tissue, and which some think, or at least we do, is the closest thing a mouse has, this more fat tissue here, is to a human like me around the waistline. This is a model, we believe, of white adipose tissue and possibly the human waistline, although some people would disagree with that. And so he went on to ask the question, does the FGFR4 deficient mice exhibit hyperlipidemia that's associated with metabolic syndrome? You got high cholesterol, you got high triglycerides that give rise to cardiovascular problems. And indeed, the answer was yes, it plays a role in control of blood lipids and adiposity. He then asked the question, does FGFR4 deficiency affect glucose metabolism or diabetes or insulin resistance, which is another parameter of metabolic syndrome? And indeed, the answer was yes. These mice are diabetic. They're resistant to, they exhibit insulin resistance and glucose intolerance. So the FGFR4 knockout, taking away FGFR4, makes the mice both high lipids, high blood lipids, and they're diabetic. Now, the next question that we wanted to ask is that, okay, we can go on with that. The next question he wanted to ask is that since FGFR4 maintains systemic lipid and glucose metabolism, it controls adiposity, does it cause the, it causes the dyslipidemic and diabetic elements of metabolic syndrome, what happens specifically in the liver? And does FGFR4 in the liver control the fatty liver that's associated with chronic obesity? And so here was a puzzling result. It was a disconnection that, to our surprise, the deficiency of FGFR4 in obese mice fed a high-fat diet actually don't have fatty liver. So in the liver, FGFR4 cured fatty liver in an obese person. However, remember, by not having FGFR4, you don't have fatty liver, you got high blood lipids. So it's a disconnection from that aspect. So we went on then to ask if it affects glucose homeostasis. This is a global knockout, so what we did, we wanted to know what is the role of specifically hepatocyte FGFR4 in the liver, since FGFR4 is knocked out in all tissues here. So what we did with that is to restore, do a rescue experiment, put back FGFR4 in the liver and see if it overcame the deficiency of knocking out FGFR4. And indeed, what we found is that the overexpression or restoration of hepatocyte FGFR4 depressed the plasma lipids, it rescued the mice from hyperlipidemia, and it alleviated the problem. However, as we showed before, it also restored the fatty liver. So it brought back the fatty liver, so this made us conclude that hepatocyte FGFR4 plays a major role in both blood lipid homeostasis and lipid metabolism in the liver. Now the next question he asked was, does the hepatocyte FGFR4, putting it back, rescue the diabetic mice? Well, the answer there, to our surprise, was no, it does not. So this means that hepatocyte FGFR4 is not sufficient to explain the diabetic effects of knocking out R4 in the whole animal. So we conclude that let me back up 1 second here, he did show that some of the basis for the hyperlipidemia in the liver was were the effects on enzymes that are going up that affect liver lipid metabolism that would explain the hyperlipidemia and the lack of fatty liver when you did knock out R4. However, what he found is that the hepatocyte FGFR4 had no effect on the rate-limiting enzymes of glucose metabolism. So this meant to us, together with this, that FGFR4 is on diabetic effects or glucose metabolism, insulin resistance, is taking place in another tissue than liver. It's indirect. And we don't know what that tissue is yet. As most of you know, if you know metabolism, it's a complicated circuit of 3 to 4 different organs. Liver, where FGFR4 is the only receptor in hepatocytes, and R4 does appear at some stages in skeletal muscle. It's not in adipocytes. So if adipocytes are involved here, it's not FGFR4. And we have found most recently, and I don't have time to tell you about that, that it's FGFR1 in adipocytes that is giving this effect, that is participating there, not R4. So what we're doing now is to do tissue-specific knockouts of FGFR4 to answer this question, which we'd like to know. So in summary, here's a summary of all this that I'm not going to go over in detail. You can read it here, and I hope if most of you can read it in the on the video if you want to go back to it. Now, how much time do I have here? Okay, 10 more minutes. So we then went on to ask another question. Does ectopic FGFR1 drive hepatocarcinogenesis? That is an observation that one sees normally here. Or does the resident FGFR4 that is a metabolic control system, is it having any effect to do on hepatocarcinogenesis? So to make a long story short, what we did is this again was Wang. He found that if we introduce in the mice ectopic FGFR1, it drives hepatocarcinogenesis in partnership with an initiator. It won't cause tumors alone, but together with DEN-induced tumors, it is a powerful promoter. and it promotes that through, this is just some of the data here. It shows bigger tumors and some of the immunohistochemistry and some of the statistical data here that shows that it accelerates. And then he found that the underlying mechanism of what FGFR1 is doing is that it is increasing proliferation but not cell death. So it's acting like a typical growth factor when it gets in the hepatocyte ectopically. It's normally not in the hepatocyte. And also he found that it increases in late stages the VEGF, and it induces VEGF and causes angiogenesis and therefore accelerates the growth of the tumors through, we believe, VEGF induction in the tumors. Now the most important question was, does a resonant receptor that has a functional role, metabolic function in the hepatocyte, is that affecting hepatocarcinogenesis? Because if that is a promoter like FGFR1, it's very dangerous to have an endocrine system that is going up and down every time you eat a meal, fat or whatever, that would be a tumor promoter. It would be very surprising to me. So indeed what we found is that in contrast to FGFR1, in the knockout mice of FGFR4, they had accelerated tumorigenesis. So this means that FGFR4, the resident FGFR4, whereas ectopic FGFR1 is a strong promoter of carcinogen-initiated hepatoma progression, R4 actually has a suppressive effect on tumorigenesis in the same conditions, which is very nice because you have a bonus there. You have your endocrine system, the metabolic control system, that is also giving a prevention effect for tumors in this case. And so I'm running out of time here, so I won't tell you the mechanism of how these opposite effects work, but we do have some ideas. R1 and R4 have different effects on some of the classical pathways for which they do act with. So to summarize that part, as I've told you here, that's the two factors are quite different. We believe we know part of the mechanism of why they're different. FGFR1 is a tumor promoter. The resident FGFR4, like Dr. Matsubara will tell you about with FGFR2 in prostate, is a tumor suppressor, which is a nice thing to know, that you're not getting initiation through a natural receptor. So the key question, and coming to the end here about all this, is how can such similar FGFR isotypes with extracellular cofactors now, and there's a new cofactor that's come into play that explains a lot of this, cause such diverse intracellular signaling endpoints and phenotypes?